Okay, so I'm going to start with a little bit of a review of the cell cycle before we get into mitosis, which will be the focus of today's mini lecture. But first, remember that the cell cycle is really in two phases: interphase, which is the time between divisions, and mitosis. And remember that interphase is separated into G1, S, and G2. During G1, protein synthesis happens, and whatever the kind of cell is, it's doing its job, making whatever proteins it normally makes. So if it's a pancreatic cell, it's busy making insulin. So it's transcribing the insulin gene and translating the insulin gene and making the insulin protein. Now during the S phase, that cell needs to make an exact copy of its DNA in preparation for when the cell is going to divide, so the new cell that results from cell division will have all the DNA it needs to make the protein, and in this case, let's say insulin. Okay, so during the S phase, DNA replication occurs. Now during G1, again, we have protein synthesis occurring, and of course, some of that time is spent with the proofreader enzymes, the DNA polymerase, making sure that no mistakes were made during DNA replication. But again, if it's a pancreatic cell, it's making insulin. If it's an intestinal lining cell, it's making lactase to break down lactose. Whatever type of cell it is building its normal proteins. And then the cell divides. So during G1 and G2, the cell is performing its normal functions. It's growing in volume and size. It's performing its normal cell functions. And during the S phase, DNA is replicating and getting ready for giving all the amount of DNA for the next phase. Now, remember that there are two shapes of DNA. There's chromosomes and chromatin. Chromatin is thread-like, and chromosomes look kind of like X's. And if you see down here in the picture of mitosis, the chromosomes are in the shape of X's, which really makes sense because an X is really easy if you have a spindle fiber to pull on those chromosomes to pull them away from each other. It makes it easy really to divide DNA if it's in the proper shape. During the rest of these phases, DNA is chromatin because if you have to get in there and transcribe one specific gene, it's much easier if it's loose and thread-like rather than tight and coiled up and ready for dividing. So when you're taking the test, if you're trying to remember, when do we see chromosomes and when do we see chromatin, if you go over to the test questions that are all about mitosis and asking you the stages of mitosis, and you'll be able to see in the pictures there are chromosomes and you can even count them, that should remind you, oh wait, these are the stages of mitosis. I'm looking at mitosis, so that's when you can see chromosomes. So again, where are we in the cell cycle? We are at the mitotic phase. Okay, and there are two parts of mitosis and cytokinesis. Cytokinesis is the division of the cytoplasm, and mitosis is the division of the nucleus. The nucleus divides first, and then the cell divides after, although there is some overlap. All right, so again, chromosomes only visible during the M phase of the cell cycle. So you may have learned about some of these phases in middle school, but the acronym that we always use is IPMAT: interphase, prophase, metaphase, anaphase, and telophase. And remember, technically, interphase is not part of mitosis. It's the time between mitosis. Some people will just refer to the entire cell cycle as the mitotic cell cycle, or as mitosis, but they really should refer to it as the mitotic cell cycle. And before we get into the specifics of what happens during each phase, I want to talk about why mitotic cell division occurs. If it is in a single-celled organism, one cell divides into two separate cells, and now you have two organisms. So that is a form of asexual reproduction. If you have a multicellular organism, like us or plants, there are three real reasons why a cell might divide. One is for growth. We all started out as one cell and now we're hundreds of trillions of cells. Another one would be repair, if you get a cut or break a bone. And also, and this is sort of similar to repair, but just replacement of cells that die from old age. There are certain cells that don't turn over very quickly, like your brain cells and your heart cells, but your cheek cells and your skin cells and all of your other cells, about every 3 months you have complete cell turnover. And I'm going to stop for a quick question. because somebody here has a question. What was your question?
I was just going to ask how long a cell lives, but you kind of answered.
How long a cell lives? It really depends on the cell type. So brain cells live a lot longer so long as we take care of them. But skin cells, because they're exposed to the air, dry out and die a lot. And actually somewhere there was statistics that 80% of the dust in your house is dead skin cells, which is kind of gross.
Not really, because like you're covered in skin.
Okay. Anyway, so the first phase, so let's get into the phases. The first phase is called prophase. Think of it as like the prologue to a book, and that will help you remember that it's in the beginning. But during prophase, the nuclear membrane disappears. This is the only time DNA is ever not technically in the nucleus, because if you're going to separate chromosomes, they have to be able to get out of the nucleus. So the nuclear membrane has to dissolve. And chromosomes become visible. Now remember, they are double-stranded because this is after DNA replication has occurred in the cell cycle. So think of middle, that's the next phase, metaphase. In metaphase, the chromosomes line up single file on the equator. And then spindle fibers, which are in this diagram, these straight lines, they're kind of like puppet strings, like marionette strings, pull apart the chromosomes. So they begin to pull apart the chromosomes and divide the sister chromatids and begin to separate them. Just keep in mind that when you're seeing diagrams of metaphase, you're always looking for chromosomes in a straight line, but they may not be vertical, because when cells divide, they're all going to be facing different directions. So sometimes it'll be at a diagonal this way, sometimes it'll be at a diagonal this way, and sometimes it could be this way. All right, the next phase is called anaphase. In this case, the sister chromatids pull apart and move towards the poles of the cell. And the cell membrane begins to pinch in. So if you see chromosomes going to the opposite sides of the cell, you've got anaphase. I want it noted that they're really not called sister chromatids once they're pulled apart. A sister chromatid is half of a double-stranded chromosome, but once they're separated, they are considered single-stranded chromosomes. Now, the next thing that happens is that the chromosomes uncoil and become chromatin. The nuclear membrane forms around each new nucleus, and the cell membrane finishes pinching in. And that is called telophase. So it goes prophase, metaphase, anaphase, telophase. And if you include interphase, it's IP-MET. Interphase, prophase, metaphase, anaphase, telophase. Now, on your unit test, we don't ask a lot of questions about the stages. We have one question where we have them all out of order and you have to put them in sequence. And even if you don't know the names, you ought to logically be able to put them in sequence based on the locations of the chromosomes if you know the idea that the chromosomes are separating to move to other cells. So, you know, what has to happen in the sequence, even if you don't know the names. And then you're going to be asked for one of the pictures to identify one of the stages. You just don't know which stage that will be. So if you don't know the stages, you really will only lose 2 points. It's not going to be a big deal. So I wouldn't spend a lot of time on the stages, but we are going to practice the stages on our mitosis lab. So by the end, the key is that you have genetically identical daughter cells, and they're now in interphase again, because remember, it is a cell cycle. So the idea is that the cells are genetically identical to the parent cell, and they're genetically identical to each other. So we're going to practice looking at a couple of pictures and identifying the stages. These are actual photographs. Now, on your test, you won't have photographs because they don't photocopy very well. They're also harder to distinguish. We always use diagrams because it's much easier to photocopy them. Now, this one is in interphase. You can't really see any chromosomes, so that's a pretty good indication that it's interphase. Now, if you look at this picture, you can start to see the chromosomes. It kind of looks like worms. And, you know, again, you really didn't know what to look for in this one until we saw that one. So if you can't see chromosomes, that's interphase. If you can begin to see chromosomes, but they're not very organized, you have prophase. Now here, this is interesting because this kind of gets at the idea that there's no real distinct beginning of one stage and end of the other stage, that these stages really flow together. So this one, some people would look at that and go, oh, that's the end of prophase, and other people would look at that and go, oh, that's the beginning of metaphase, and most scientists would call that prometaphase. I would never give you one like that on the test. When we give you diagrams on the test, it will be obviously like smack dab in the middle of the stage so that it's pretty clear. This one would be metaphase, and what I like about this photograph is that even though the chromosomes are in a straight line, they're not vertically like they are in most diagrams, because in a real cell, they're going to line up on the equator, but the equator may be in different directions. Now the green you see in here are the spindle fibers, whose job it is to pull those apart in the next phase. And this is anaphase. You can see the chromosomes, which are dyed blue, are being pulled apart. And here we have telophase, and you can see cytokinesis is occurring. Cytokinesis is actually the division of the cytoplasm. Now, which stage do you think this one is in? In this case, it helps to know that the chromosomes are yellow in this particular stain. So what stage do you think it's in? The chromosomes are being pulled apart. It's anaphase. So what is cytokinesis? Cytokinesis is the division of the cytoplasm, and it begins sort of during anaphase and continues during telophase. So in an animal cell, cytokinesis is much simpler because all you have to do is pinch in the cell membrane, because remember, the phospholipid bilayers are very flexible. When we did the bubble lab, we were actually kind of playing around with that. So in animal cells, the cell membrane pinches in. This is also true for protozoans who have no cell wall. Most cells have cell walls, but the protozoans, like the amoeba, paramecium, and euglena, which we studied during the cell unit, and you had them on your flashcards when you were learning about cilia, flagella, and pseudopodia. Those guys do not have a cell wall, and so they divide just the way an animal cell would. But anything with a cell wall can't just pinch in because a cell wall is rigid. If you have a plant cell or an algae cell, it's made out of cellulose, and we already know cellulose is very hard, and all of the wood that we use for building is very rigid. So what happens is a cell plate forms in between the two cells, and then cellulose gets deposited on the cell plate. So in plant cells, the cell wall prevents the cell membrane from pinching in because it's rigid, and instead a cell plate forms. So in this photograph, you can actually see the cell plate are like little vesicles, or like little phospholipid bubbles, and then at a certain point, cellulose, as it is built, will be deposited along that line. And if you guessed that the two new cells will be smaller, that's certainly true, but they will eventually grow in size. Now, this photograph is showing an animal cell dividing, and it's pinching in. And it's called a cleavage furrow because this is, in all likelihood, an egg cell that divides. And when egg cells divide, they don't have time to grow in between mitotic cell divisions. And so it's called referred to as cleavage. So in some organisms, cytokinesis isn't exactly equal. And so usually with normal mitotic division and cytokinesis, you get two equally sized cells, but sometimes you don't. And when you don't, it's called budding. And yeast, which are a single-celled fungi or fungus, are the prime example of the organisms that are associated with budding. So this would be your bud, and eventually these small cells would grow in size to reach, equally the same size as the bigger cells. So what if you don't have a nucleus? Your nucleus can't divide if you don't have one. So in prokaryotes, they don't call it mitosis because mitosis is technically the division of the nucleus. But what happens is largely the same. Now remember, prokaryotes are bacteria. All bacteria are prokaryotes. They have DNA, but instead of being in linear chromosomes like ours, they have one loop. And what happens is that loop replicates, and then the cell divides. So in terms of the cell cycle and the amount of DNA, and DNA replication and then the cell dividing, it's largely the same, except for, you only have one chromosome that's dividing, and it has a special name, and that name is binary fission, and that literally means dividing in half. Sometimes if you go on the internet, they will refer to any single cell dividing as binary fission rather than just limiting it to prokaryotes. But if you are a eukaryote and you are single-celled and you divide, you do have a nucleus, so you can have mitotic cell division. So we can pause here for a second. I'm not going to pause the video because then I'll have to splice it together. but you can pause here and try and answer these questions. So the 4 phases of mitosis in order would be prophase, metaphase, anaphase, and telophase. Now, if we're trying to identify which cell is which, let's first find prophase. Remember that we said in prophase, we're looking for chromosomes. So you can see chromosomes, but they're not organized. They're just kind of randomly distributed throughout the nucleus. So that would be the third one from the left. Now in metaphase, think meta, middle. So in metaphase, they're lined up in a straight line. So that would be the first one on the left. And then in anaphase, they're beginning to separate. Yep. And then in telophase, you're starting to see that there are 2 new cells and the new nuclear membrane is starting to form, and you can still see chromosomes, but they're becoming chromatin. So that would obviously be the second picture. So in which cell type does cytokinesis occur by pinching in, plant or animal?
Animal.
Yep. And a cell plate forms in
Plant
cells because of the rigid
cell walls.
All right. End of video.
Wow, you're cool.